So today we will be reading Staging of the estrus cycle and induction of estrus in the experimental rodents, an update. So we do this for securing there are advantages that include secure handling of subject. We learn to secure how to handle the subjects in a secure manner, elimination of attribution from the study, control environmental experiment conditions are given for this experiment. So commonly animals used are mice, rats and rabbits. Small rodents like mice and rats are more preferred to the large animals like rabbit, dog, cat, pigs and monkeys. This and in reproductive function research, particularly those involving the use of female animals, mice and rats are commonly used due to their well characterized estrus cycle and secure handling. The short and precise length of estrus in these rodents also make them very suitable. Determining the estrus phase is essential for selecting female animals that will mate when paired with the male counterpart to achieve timed pregnancy, or tracking estrus for variable that may affect the research. Assessment of the estrus cycle in the experimental animals is a useful measure for the integrity of the hypothalamic, pituitary and ovarian axis and the functioning reproductive status of the female reproductive system. It can also be used to investigate the effect of drugs and chemicals on the reproductive function, frequently expressed as the disruption in the typical morphology, cytology and histology of reproductive organs and alteration during on the duration of particular phase of the estrus cycle. The estrus cycle, the reproductive cycle of rodents, okay, it is similar to the menstrual cycle in humans. There are four phases: proestrus, estrus, metaestrus and diestrus, and last for 4 to 5 days. So let us see the table 1, the estrus phase of rats and mice. So proestrus lasts for 14 hours and less than 24 for mice. Estrus 24 to 48 for rats and 12 to 48 for mice. Metaestrus 6 to 8 for rats and 8 to 24 for mice. Diestrus 48 to 72 for rats and 48 to 72 hours for mice. So the reproductive organ and estrus cycles of the mice commence about 26 days after the birth with the opening of the vagina, which is about 10 days before the vaginal cornification. The apoptosis mediated vaginal opening is an essential secondary character in mice, which is used as a predator of puberty. The vaginal unfolding is associated with an increase in estradiol concentration. So vaginal opening when the estradiol concentration is increasing. In rat, a vaginal opening occurs during the first ovulation. Okay. In female rats, puberty is preceded by a pulsatile release of luteinizing hormone after the fourth postnatal week, approximately 30 days. This period is the anestrus and can occur about 8 to 9 days before the first proestrus. The first proestrus, estrus, metaestrus, diestrus then follow. Metaestrus only occur in absence of conception. So here they are showing the similarities. So first they are talking about humans. They have 3 phases: menstrual, proliferating and secretory. So for proliferatory, it's also called follicular, and secretory is also called luteal. What happens in proliferatory? The high estrogen levels are seen, Okay, and in secretory, high progesterone levels are seen. They both together are called ovulatory phase. Okay, here ovulation occurs following the LH surge. This is a cycle of 28 days. Now in rodents, it is a cycle of 4 to 5 days. It has 4 stages: proestrus, estrus, metaestrus, and diestrus. Proestrus is similar to follicular stage. Metaestrus and diestrus, it is similar to the early and late secretory stage. This secretory stage. Okay, so what happens in proestrus? There is a rise in estradiol and prolactin, which rises the LH and FSH. Okay, now when the peak of FHS is reached, decrease in estradiol is seen, and when a decrease in estradiol happens, ovulations take place. Ovulation take place means now we have reached the estrus stage. So if conception occurs, then metaestrus will not follow. If conception does not occur, then metaestrus will follow. So this is what is said here. Historically, estrus was first used by Heath. This was Latin adaptation of the Greek word oestrus, which means sexual season, fire, frenzy, string, or madness. Proestrus is the pre-predatory stage of the animal coming into heat. Metaestrus is the brief period characterized by the decline of corpus luteum function in the absence of the conception, when the activities of the reproductive organ gradually subside. Diestrus, which is the period of the short rest during the breeding season, and estrus, which is the non-breeding period when the reproductive organ are in question. You can read the more. I don't want to read it right now. So here is the description of the all different phases. So during proestrus, vagina is gaping and the tissue are moist and reddish pink. There is this. Okay, proestrus, vagina is gaping and tissues are moist and reddish pink. There are numerous longitudinal folds and striations visible on the dorsal and ventral lip. Estrus, vaginal appears similar to that seen in proestrus, but the tissues are lighter pink and less moist. Similar to the proestrus. The striations are more prominent. Metaestrus 1 and metaestrus 2. The picture only shows metaestrus, not a differentiation. Vagina tissues are pale and dry, and the dorsal lip is not as oedemestrous as in estrus. Vagina appears similar to that seen in metaestrus 1, but the lip is less oedemestrous and has receded. Whitish cellular debris may line the inner wall and partially fill the vagina. Metaestrus, whitish is seen here. Okay, vagina is moist and has small opening, and the tissue are bluish purple. So I cannot see, little bit maybe bluish purple, but I cannot see it clearly. So there are different techniques that are talked about. Okay, so there is vaginal cytology, histological examination of the reproductive organ, vaginal wall impedance, and the urine biochemistry. So different are talked about. This we can just look at the vaginal opening at different time period and decide. Okay, appearance of vagina in different phases of estrus cycle. Okay, this is one of the way. Second is making a smear. So In a pipette, saline is used, okay, and the liquid then they take and slowly release the liquid into the vaginal and draw it into the tip. This should be repeated 4 to 5 times in the same sterile latex. Do not penetrate the vaginal orifice. The liquid containing few drops of the cell suspension is after introduced in the glass slide, air dried and contained. Okay, we are making a smear, and then we'll put this slide in the 1% crystal violet stain. Other than this, other stains are also there. Romanovsky type stains as modified Wrights and Wrights Gemsa, tulip green blue O. The slide is an later covered slip and then seen under the 200x micros magnification. So it is seen in proestrus. Neutrophils are 0 to positive presence, small nucleated. Okay, there's other way. You can just take a swab and then make a slide and see it. Okay, so in a mouse and a rat, there are 4 types of cells we are basically seeing. Neutrophils, small nucleated epithelial, large nucleated epithelial, anucleated, keratinized. Okay, so basically only 3 are there: small and large nucleated epithelial. Okay, so during diestress, mostly leukocytes are what we are seeing. If it is a late diestress, we are seeing nucleated epithelial cells too. Okay, in proestrus, nucleated epithelial cells are seen, but if it is a late proestrus, we can see cornified epithelial cells too. Okay, so in estrus, cornified epithelial cells are seen and nucleated epithelial are seen if it is early estrus. Now in metastrus, all 3 are seen. Similarly in rats, we'll see in diestress leukocytes. In late diestress, nucleated epithelial cells are seen. In proestrus, nucleated epithelial cells are seen, but in late proestrus, cornified epithelial are also seen. Estrus, cornified epithelials are seen, but in early or late, a little bit of nucleated epithelial are also seen. But in metastress, all 3 are seen. This is what it is written here. Okay, these clusters are here. Okay, so how do they look? Yes, a cell and nucleated, like this, nucleated epithelial. See, look at them, nucleated epithelial. You can see the cell and nucleus inside. This is in estrus phase. See, cornified epithelial. We cannot, these are sharp ended and they do not have nucleus. You cannot see nucleus in them. So there is nucleated epithelial here, this cornified here, nucleated epithelial here. Now let us see some leukocytes. These are leukocytes. Okay, this is nucleated epithelial. Cell is there and nucleus is there. So here other things, histology is told. Okay, proestrus, how do they look during proestrus? Vaginal, mid or late, early uterus, uterus. What happened during estrus? What happened during metastrus? it is gone in very deep. Okay, so that's all. Summing up, it is worth to know that the various techniques can determine the phase of estrous cycle. Careful selection of the method employed is very essential. Vaginal smear cytology remains as the standard in life animals upon which other methods are verified. Although histological examinations of reproductive organs are as specific and reliable as vaginal smear and cytology, it is not useful in the live animals. Improvement of the available techniques to enhance these reliability and specificity is pertinent. Studies to achieve benchmark values of vaginal wall impedance and urine biochemical parameters at different phases of estrus is commonly used. Experimental animals is also essential. So that's all for today. Thank you. Bye bye.